Okay, um, so Happy New Year, everybody, and thank you for everybody who's joining or will join or watching this recorded. I'm Mariam, the Science Communication and Marketing Officer at GenomeArc. Today is our first talk in 2024. It's a very special talk, and we have a very special guest today, Dr. Karolina Kobus. Dr. Kobus is the head of the Molecular Genomics and Precision Medicine Laboratory in ExpressMed Labs in Bahrain. She is a molecular geneticist and precision medicine consultant with extensive experience in the field. Today, she will be discussing genetic testing in oncology. It's a very exciting perspective from an expert in the field, and we're very excited to learn more about it today. Thank you very much for joining us today, Dr. Kobus.
Thank you, Mariam. Good morning. Thank you, Mariam, for your kind invitation. I'm very happy and glad to be there with you. So today, as you mentioned, we will talk about the importance of genetic testing in oncology field. So cancer is an universal public health problem and its leading cause of death worldwide, accounting for estimated 9.6 million deaths in 2018. And what we observe is the cancer cases are raised not only in Western world but also in Arab world. So it's a global healthcare problem, and with the projections that by 2030 the increase the prevalence of cancers will be high as 1.8 in Arab world. Nevertheless, if you see the specific cancer studies in Arab world, like Gulf Cooperation Council, counted the colorectal cancer, for example, and reported already 2.7-fold increase in recent years in both genders for colorectal cancer. Same or similar situation is for breast cancer. So many patients are diagnosed with cancer in Arab world at a younger age compared to Western countries. So when we see the colorectal cancer, for example, it affects younger population in UAE or other countries, Arab countries, and when you see the percentage, almost 30% of cases are below 50 years. 22, 23% cases, cancer cases below age of 40. In Egypt, the proportion of young colorectal cancer cases is reaching even 36%. So when you compare that to Western countries, where 90% of cases occur at age of 55. So we need to be aware about that differences, especially that nowadays are genomic study and services going on in that particular region. So when we see breast cancer, as I mentioned, that also many Arab women are diagnosed with that type of cancer, not only breast cancer but ovarian cancer as well, below age of 40 in our region. So more Arab women and patients are diagnosed with larger tumors, positive lymph nodes, and metastatic disease compared to Western countries, compared to other Caucasian or Afro-American ethnicities. So when you see the statistics, even taken from Bahrain, the breast cancer incidence increased or doubled within the last 20 years. So when we see the current breast cancer or cancer screening recommendations in our region, usually they are copy-paste from Western countries like United States and UK. They just follow the worldwide standards. But we have challenges here. So, for example, in many Arab countries, still there is some lack of insurance and coverage for genomic, preventive, predictive genetic testing. Like BRCA1, BRCA2, genetic testing is not covered in many countries by insurance. So it's not the same situation as in European countries, for example. We are also facing lack of information about the family illness history. We have also here, and this is also somehow related, and we should not forget the high rate of consanguinity and kind of geographical isolates, which widely and greatly affected or impacted the genetic makeup of the entire region. So what would be the solutions? Definitely rapid detection and diagnosis of cancers are urgently required. So the presence of the genomic laboratories and services being offered locally is very important. Need for more research, particular cancer profiling, studies for Arab population is very important. And of course, based on that data, we should start adjusting current diagnostic and screening programs to the needs of Middle Eastern populations. So as we see, overall, we can categorize the cancers as a sporadic, and we count them as for, let's say, 70-75%. We have inherited type of cancers like breast cancer or ovarian cancer or Lynch syndrome. It's counted for 5 to 10% of diagnosed cases, but less understood pattern in Arab world, what we've seen in different countries, we can call it familiar cancers. It's counted for 25% of cases where there is patient is having very clear family history of the disease, but there is no clear pattern that is consistent with any of known inherited syndrome. That's why there is need for more research, particularly studies related to comprehensive cancer profiling, which will give us understanding of molecular pathways involved in that disease, in that particular ethnicity. So recent technological advances, especially in next generation sequencing, but not only, definitely significantly improve the diagnostic yield for both, for the disorders where disease genes were unknown before or genetic testing was not offered. So provided completely new innovative cell-free or single-cell diagnostic applications such as non-invasive prenatal testing, brain transplantation genetic testing like PGTA, PGTM, or liquid biopsy testing where we can catch circulating cancer cell and make a tumor profiling in non-invasive matter. So, and open new revolutionary chapter for the precision medicine, which is transforming the landscape of the disease diagnosis and treatment. So definitely we see the shift that we see that new genomics applications and technology and softwares, AI, machine learning solutions, giving us much faster, smarter, and more efficient diagnosis. So personalized medicine is definitely emerging approach for disease diagnosis, treatment, and prevention, as we discussed. And oncology is one of the most important field of that personalized medicine, but not only. Personalized medicine is also applied widely for diabetic and cardiovascular diseases. And moving away from one size fits all model to the more tailored and customized solutions. Why is that? Because actually cancer is not a monolithic illness. It's different diseases. It's very heterogenetic disease. That's why each patient's journey towards recovery is unique. And that's why some patients do respond to the specific treatment, while others not. So we need to be aware about that. And on the top, due to family history, some people are genetically at higher risk to develop certain type of cancer. So it's very complex issue. And that's why we have that shift paradigmic where we have that tumor heterogeneity was studied extensively, and it emphasized the need of tailored therapeutic strategies based on unique genetic composition of the tumor, molecular composition of the tumor, and clinical characteristic of each patient. So as we see, what we've seen already, only in 2023, so last year, FDA approved more than 55 new drugs. So the new tools are continuously being developed, and there is race between different pharmaceutical companies to make cancer treatment more efficient and to create and identify the new biomarkers. So as we see from the genomics lab perspective, the workflow it's very complex and very comprehensive. So we have tissue testing and we have blood testing. Tissue testing usually it's applied for solid cancers and to identify the somatic mutation. It's for diagnosis and prognosis purposes. And then we have also blood testing, historically, to identify the germline mutations for hereditary cancers. But recently, we have also liquid biopsy and cell-free DNA approaches to identify the composition of the tumor in non-invasive manner. One can see here different machines can be applied for the comprehensive genomic assessment. We have on the left real-time PCR, the classical approach. We have then digital real-time PCR, which is widely used nowadays for liquid biopsy testing. Then we have Sanger sequencing approach, which is also a bit historical approach, and then followed by a next generation sequencing. So if we see involvement of the personalized oncogenomics strategy and services from the laboratory perspective, we can divide that in different generations. So the first generation was targeting single gene testing. We were doing that either by real-time PCR or by Sanger sequencing. Both strategies, if we consider the paradigm of and the idea and agree that the tumor and cancer are heterogenetic biological phenomena, definitely it was not effective approach. And even from the laboratory perspective, the real-time PCR or Sanger sequencing, it's also very DNA or material consuming. And if we talk about the biopsy testing or tissue testing, we don't have much material for the genetic investigation. So we would like to have a much better comprehensive approach where out of certain amount of DNA we can test as much biomarkers, as much targets as possible, especially that we need to remember that the tissue, the part of the biopsy, it's not only given for the genetic investigation, it's split between anatomical pathology and genomic departments, and it's given to the anatomical pathologists for the immunohisto and histopathological investigations. So it's precious material, and we need to be very careful how we are going to utilize that DNA or RNA. So the second generation and third generation is already addressing and overcoming that issue of single gene testing. Here we are implementing or incorporating already a next generation sequencing technology. In the second generation, a very popular approach was based on small cancer-specific panels. They were having 5 genes or 10 genes or up to 20 genes specific to the certain cancer. Let's say 20 gene panel related to breast and ovarian cancers. We shift that paradigm yet again, and we decided, because at the end, let's say KRAS gene, is KRAS gene related only to the colorectal cancer, or might be also important biomarker for lung cancer or maybe for thyroid cancer? That's why there was yet another shift, and we called it at like third generation, where we started doing the small comprehensive cancer panels, where there was 52 gene panels and also copy number variants and fusion drivers were needed. So then we moved forward and started fourth and fifth generation of the screening, where we implemented the big NGS panels with 500 plus genes, fusions, HRDs, tumor mutation burden, and MSIs. So there are some agnostic biomarkers which are applied and important for any type of cancers. So because of that big data and enormous informations, the fifth generations was important to be able to analyze the data and generate meaningful reports by integrating artificial intelligence and machine learning into that genomic data analysis. Without AI, without softwares, without machine learning algorithm, we will not be able to cope and provide all that meaningful information to the clinicians, because it's not only about the finding the pathogenic variant or genomic signatures, but also physicians and patients want to know the therapies associated with that variant or available clinical trials which are happening right now worldwide. So when we see that race, and when we see that the amount of biomarkers are increasing, and when we see also that pharma companies are providing so many new therapeutic and personalized therapies for the cancer treatment, how we need to somehow also cope in a professional and careful manner in the laboratory field. That's why the new assays which are related with the new biomarkers required validation, verification, assay regulations, pre-analytical QC steps, being part of the proficiency testing and constant training in terms of the results interpretations, because we need to remember that biomarkers can be checked on different levels, not only on the genomic level, but also on the proteomic level. So we have FISH stainings, we have immunohistostainings, and we have genomics. And that pre-analytical steps and part of validation and verification it's important to be sure that we are not generating any false negative or false positive results. So now let's move to the cases. That cases will show multi-omics approach for solid cancer or breast cancer testing and diagnostic from the laboratory perspective, where we share the genomics department and anatomical pathology department together in order to provide comprehensive assessment of the tissue or blood of the particular patient. So the first case, it was the metastatic colon cancer, and we applied the NGS 52 cancer-related genes for that case, and we identify KRAS mutation. That KRAS mutation is well known, well described. It's classified, the variant is classified as pathogenic. No surprise, as 30 to 40% of colon cancer carry the KRAS mutations. And we need to remember that KRAS mutations are associated with poor survival and increased tumor aggressiveness compared to wild type KRAS cases. Here, because we discussed how important is to cross-validate the result, that time that case was one of the first case done in our laboratory. So we cross-validate the NGS result by alternative assay, single gene assay coming from done by real-time PCR, and real-time PCR approach confirmed that finding. The next case, it was a very young man, 46 years old, with lung cancer, and clinicians order PD-L1 marker to be stained by immunohisto, EGFR, BRAF, and ALK fusion gene. The case was negative for EGFR and BRAF. It was applied the next generation sequencing approach for that case. Yet again, 52 gene panel, for, which is checking the DNA and also RNA. That's why we were able to identify and detect the ALK fusion variant. And the physicians were very happy about that, because that ALK fusion genes currently are very important tumor driver information for the lung cancer patients, and providing very sufficient and very effective targeted therapies, especially for young non-smoking patients, where EGFR or other gene mutations are absent. And there was also PD-L1 staining positive for that case. So in addition, that kind of patient is also eligible for immunotherapy treatment. Here we will move for multi-omics approach for breast cancer testing. So first line test, it's not the genetic testing for that kind of tumors, because 80% and 65% of that kind of cases can be solved by immunohistostainings against ER or PR biomarkers, which are hormone receptor markers. If they are negative, the second line test is HER2 stainings, and to be done either by immunohisto followed by FISH. And this type of cancers, the HER2 positive cancers, counted for 20% of the cases. They are aggressive and fast growing tumors. Nevertheless, we need to remember that there are some cases, 15 to 20% of cases, where these three receptors, ER, PR, and HER2, are negative, and then it's classified as triple negative breast cancer cases. They are considered as poor prognosis and suffering from lack of effective targeted treatment. Often target doctors often offering them surgery, chemotherapy, or radiation. But we should also remember that many triple negative cancer cases might be associated with BRCA1, BRCA2 genes. That's why in our laboratory, when we see that kind of cases where HER2, ER, PR biomarkers were negative on immunohisto or FISH level, we do recommend breast cancer genetic testing for that cases. And here we have two nice examples where BRCA mutation were identified in that triple negative breast cancer patients, and PARP inhibitors offered to that patients, and which definitely improves overall survival. And very important, that kind of cases here we identify on somatic level. And yet again, here I will share one more case of 36-year-old woman with triple negative breast cancer, and here we were testing the tissue. So we identified the BRCA mutation, BRCA variant on the somatic level. But as you see, the mutant allele percentage, it was 41%. It gave us very high indication that because of the allele percentage, that what we seen in the tissue might be the germline variant actually. And the age of the woman, of the patient, also is strong indication that that breast cancer, it's might be the inherited cancer in fact. So we requested the referral doctors and the hospital to send us blood of that patient, and we identified the germline. We confirmed that the BRCA mutation, which was seen on the tissue, it was confirmed by the germline investigation, and the variant was found in blood. And definitely that kind of patient and her entire family can benefit out of that study, out of that report, because other family members can be offered the genetic testing, the BRCA1 mutation genetic testing in terms of risk assessments and prevention. Here we will move to yet another biomarker. This is before that biomarker was related to colorectal cancer and Lynch syndrome. Nowadays it's expanded and became a agnostic, tissue agnostic biomarker for other solid tumors, not only for colorectal cancer tumors, and patients who suffer from microsatellite instability can be like offered and be beneficial for and referred for immunotherapeutic inhibitors treatment. So here, historically, MSI was done by and performed by PCR, followed by capillary electrophoresis, and it's like fragment analysis done by genetic analyzer. There are different assays for MSI, can be focused on 5 biomarkers or up to 13 markers for that particular assay. Nowadays, there is yet again a movement where instead doing the PCR and genetic electrophoresis for MSI studies, we can apply NGS applications based on big 500 gene panels, fusions, where we can calculate MSI, HRDs, and also tumor mutation burden. So all in one, we have the comprehensive assessment. So here we have yet another case. It's gastric cancer with very poor prognosis because it's frequently diagnosed at advanced stage. And although despite aggressive treatments, 50% of cases are counted to develop the recurrent disease. This particular case was sent to our laboratory for HER2, PD-L1, and MSI testing. This particular case, we applied the capillary electrophoresis assay for MSI, and we identified the microsatellite instability high biomarker, and we also make a FISH for HER2 gene. It was negative, and as you can see, it was positive for PD-L1. That's why definitely that kind of cases can benefit from immunotherapeutic targeted therapies. The last case I would like to show, it's the 44-year-old man with colorectal cancer, where we apply the comprehensive assay, which we already discussed, where we do everything in one. So we check MSI, we check tumor mutation burden, we check the DNA and RNA fusion drivers, and so on. And that's very interesting case because we identify MSI unstable for colorectal cancer. So as per the guidance, if you have MSI high and for colorectal cancer, there is likely the patient is likely to have a Lynch syndrome. So, and as you can see in the table, one of the gene MLH1 was identified on the tissue, And yet again, you see the percentage of the mutant allele, it's 30%. So it's giving indication that if we take the blood of that patient, that MLH1 pathogenic variant will be seen on germline level as well. So that's a beauty of that fourth, fifth generation of genetic approach and assays where we do everything in one, and patients and the doctors getting all informative decision in one. Here it's very important to mention, because the last cases, the five or six previous cases were positively resolved. We found the pathogenic variants and gave some hope and chances to provide the targeted therapies for that patients. But we need to remember that 25% of the cases in the Middle Eastern regions have positive family history, but there is no clear pattern that is consistent with any known inherited syndrome. So that kind of cases are being sent for cancer testing or NGS specific panel testing, and coming out either with false negative results, or many of them, up to even 60% of the cases, coming with the variants of uncertain significant. So we are expecting a lot of VUS, we are seeing a lot of VUS, and we are seeing a lot of GUS. Means if we apply exome sequencing or genome sequencing for that cases, we will find uncertain variants or genes of uncertain function. So how to solve that dilemma? So definitely national genome projects, national cancer genome projects are key to understand and solve that kind of cases, followed by, of course, functional analysis. So why we have actually that situation, why we face so many VUS and GUS situations and results in our region? Because we need to remember that the current reference genome is derived from single Caucasian individual, so does not represent the global diversity, and the Arab population in particular, it's underrepresented in that matter. So that's why we are facing, as a consequence, false negative or false positive results. And this last year in March, that was published the Human Pangenome project and data, and it was representing 47 individuals, and none of them were coming from Arab population. Nevertheless, there are beautiful genome projects going on in our region, in UAE, in Qatar, in Saudi Arabia, in Bahrain, and thanks to that kind of initiatives, luckily we will fill the gap and put and generate the data coming from the Arab patients and ethnicities, and include them to the genome and the referral genome reference. So definitely what's I can see also personally what's the importance of the genomic initiatives in the Middle East. as a human, we have 20,000 genes, and as for now, more than 50% of that genes, so we talk about almost or roughly 10,000 genes, are without any known function. So I do believe that the Middle Eastern genome projects and pan-genome projects which are going on, and functional genome initiatives definitely will provide very impactful insight in worldwide understanding of functioning of that unknown genes. So as a summary, the local genome and cancer genomes initiative, they will fill the gap which we are currently facing. It will definitely improve the diagnostic yield. It will reduce the false positive or false negative reports. It will definitely decrease amount of VUS and GUS results, and clarify the reason behind the higher genetic disease incidence in GCC countries. Yeah, and that would be my last slide, and thank you for being with me today, and I'm happy to take any questions.
Thank you. Thank you very much, Dr. Kobus. It was a very interesting talk. It was really interesting to see all the different tests and analyses from different aspects that are used to profile these cancer cases. Especially the last one with that combined all the analyses together. The last case. So maybe I can ask a question. You've mentioned that there's a huge role in AI and machine learning that will help in the therapeutics and the sort of precision kind of targeting for cancer. How do you think this will be? How do you think this will impact really with all these analyses? Well, what will it solve the most? Like considering the problems you might face with these analyses, whether it's not detecting certain mutations, or how do you think automation, machine learning, AI would help in these stuff?
Thank you, Mariam, for a very nice question. Actually, AI and machine learning approaches are being applied on different levels in our field. As you mentioned, it's applied for finding and searching for the new biomarkers. So that's a different approach. And because they start applying or incorporating AI and machine learning algorithms that's why we see that kind of dramatic, rapid boost and change and increase of finding new targets and biomarkers recently. As I share in one of my slides, only in 2023 there was 56 FDA-approved new drug targets. So it's amazing discoveries, and it definitely will help patients, but we need also be aligned with that race, because as genomic laboratories, as anatomical pathology laboratories, if we are seeing, let's say, every month the new biomarkers coming out, and there is new therapeutic coming out, we need to cope with this as well, because if we will be in a hurry, If we will be under pressure of implementing more and more new biomarkers, we might lose quality, we might lose efficiency and sensitivity of that assays. So that's why AI is also helping us and softwares and machine learning, helping us to cope with data interpretation and data analysis. Because as you said, the last case, imagine if we have that kind of data being generated without ability of having softwares and AI and machine learning application, it will take days or weeks to generate the meaningful report. And as a human, we might also overlook certain variants or metrics. So AI is applied on both sides to discover and to find out the new biomarkers, but on the other hand, also it's helping us to deal with that enormous amount of data in a professional manner.
Yes, definitely. I completely agree with you. So not only it will increase the amount of data being interpreted and the insights we can get, but also the pace at which we provide this to the patient. So following up on that, how important, in your opinion, how important is it to do analysis in a time-constrained manner? Like how much does it affect patients in terms of treatment or in terms of prognosis or diagnosis?
So definitely, I'm a big fan of doing the genetic testing, especially diagnostic genetic testing locally. So availability of the genomics laboratories should be, I would say, in the country. Outsourcing that kind of tests affect the turnaround time, and also, as we seen, each ethnicity, each community has a different genomic makeup. So if we, let's say, do outsource genetic testing Arab patients to United States or to Sweden or to European countries, their algorithm, their softwares, their workflows might not be adjusted perfectly to the reality and genetic composition of high consanguinity tribal communities. So that's always giving that kind of risk of generating false negative or uncertain results. And one more thing regarding the machine learning and AI. It's very beautiful idea, but the best software, the best application, if it's applied the wrong control, it will give us the wrong outcome. So I do believe that, especially in our region, we should start from the beginning, create proper control genome reference, and then moved forward and start applying that fancy tools and applications. Otherwise, it will generate false outcomes.
Yes, yes, definitely. So again, it comes back to the different populations and the heterogeneity and how to represent that accurately. Okay.
Absolutely.
Yes. Okay. Thank you so much for joining us today, Dr. Kobus. It was a very interesting talk, and you've really shown different perspectives on how cancer cases are analyzed and the different kind of omics approaches that go into it. Thank you very much for joining us today.
Thank you so much.